All right, you guys ready to hear the quiz answers? I was laughing at myself earlier because I wanted to do like a raise of hands of people that weren't here back from break, and it just like dawned on me like, Marlon, you stupid. They have people that are not back from break yet raise their hands. Anyway, I just had a moment to myself. I can laugh at myself. All right, you guys ready for the quiz answers? All right, so you guys know the answers. You guys are gonna give me the answers. Anatomy is the study of structure, while physiology is the study of function. No, I already gave you guys the answer. It's that type of Thursday.
That type.
Yeah. So if you only got one of them right, then I will give you partial credits. You get 1 point. A condition of equilibrium in the body's internal environment. Homeostasis. Feedback mechanism that reinforces a change. Positive feedback. Isn't that the one I gave you guys?
Yeah.
Right? Okay. Receptor is also known as the blank pathway. Afferent, yes, because the receptor is receiving the information. So that's information going into the control center. This form of energy is needed to start a reaction.
Activation.
I can tell by that you guys weren't too confident about that answer. Activation energy. That's the amount of energy that you need to invest in order for a reaction to take place. And if you remember that when I said, remember this slide, and I didn't mean remember this slide just because you're going to be seeing it a little bit later on, but remember this slide because it was going to be on the quiz.
What chapter was that?
What's that?
What chapter was that?
Somewhere in the book.
The chapter first, the first.
Oh, what lecture was that?
Yeah.
That was that was Tuesday's lecture.
Oh, okay.
Oh no, I'm sorry. That was no, that was last Thursday's lecture. That was our biochemistry lecture. Where am I? This element is needed to create the backbone for organic compounds. Carbon. Organic molecule that is the source of energy currency. ATP. If you put glucose, I'll give it to you. Okay, because I know people, I've seen people put glucose on there. If I thumb through it, I'll see quite a bit of glucose in there. If I were to say the organic molecule that is the energy currency, that would have been maybe a little bit clearer, and that's what I meant, ATP, adenosine triphosphate. That's what we're going to be talking about today when we get into our cellular respiration lecture. This class of lipids that have both a polar and non-polar side. Phospholipids. If you said phosphate lipids, it's okay. But as long as you gave that, if you called it a lipid, I'm not giving you any points. If you called it something with phospho in it, that's okay. If you misspelled it, that's okay. I get the main idea. I'm not knocking points for spelling. That's perfect. Movement of water in and out of the cell is referred to osmosis. And lastly, what was the question? Ions require blank in order to cross the cell membrane. Channel. Right? So if you put carrier, that was for the bigger molecules. We're talking about the carriers today, specifically the glucose carrier. We'll be talking about channels in our neuron lecture, our neurophysiology lecture next week. So you'll be seeing these terms over and over again. Of course, naturally the question of, does everything that's covered on today's quiz, do we have to remember any of that? Well, I will be making references as we go through what things you do have to remember. Remember what I said when we first started this class, that we're building on top of that stuff. So don't forget everything, but I will be making references. Remember lecture 2, remember lecture 3, this is what we talked about. Those are going to be the important things. But the things that I mentioned before, mitosis, meiosis, do I care about independent assortment? Do I care about DNA replication? Do I care about those? Nah, as long as you get the main point of why we would be splitting cells. If you get the idea of ramping up the production of proteins requires DNA transcription, transcribing that DNA into RNA and translating that RNA to a protein. That's how we're making proteins. So now that you get the idea of genes from the DNA creating proteins down the line, now you get the idea of how we're going to start producing neurotransmitters. You're going to start understanding the concept of what I meant by upregulation. This is activating a gene to start making a protein. So you start getting those ideas. We'll be talking about enzymes and how we turn on enzymes, how we turn off enzymes. So you'll understand that process. We'll talk more about these types of enzymes as we go through. So that serves a foundation. Again, I know that was a lot of stuff to remember for the first 3 weeks, but that stuff will not be covered in lecture exam 1. Okay? Quick announcement for those that are still trying to get added to the class. there are permission codes that I will give to you. So see me before you leave class today. There's a transition in our systems where we used to give out ad slips. I don't know if you guys have taken classes here before. Instructors used to give an ad slip where I would sign, I would date, give you the slip, and you take that over to admissions. This new system, I'm required to send you, to give you a code. It's like a 6, 7, 8-digit code. And apparently that code isn't something that you use to enroll in the class. I think you still physically need to give it. So I apologize if you're only here Tuesdays and Thursdays, but you will have to come back to admissions office to hand that code back in. Good news is you have till Sunday to get it done. So I will give you the code, I'll email you the code. That's only your code, so don't share that with anybody else, but that's your code to get into this class. And that's really it. Otherwise, I'm not taking any more of attendance. I'm just going to mark off those that actually never showed up. That's perfectly okay. I'm dropping a couple people from the class because they never showed up, and then that should give us should round us off to about 30, 32 people, and we're set. So you guys are in it for the long haul. All right, you guys with me?
Yeah.
Cool. All right, so what we're going to do is we're going to go into our first lecture. And yes, today we do have 2 lectures to go over, but I do plan to kind of speed that up so that we can get into so we can get into our tissue lecture and move into our next segue, which we'll be talking about next week. So now that you have the foundation, we've talked about some of the elements, we've talked about our molecules. You know by now our 4 main macromolecules, and we've talked about the cellular structures. You're now familiar with the organelles. You're familiar with what else did we talk about? You're familiar with the enzymes, how enzymes work. What we're going to do is we're going to start to tie that in and go into our first lecture, our cellular physiology. And with that, we're going to look at respiration. And one of the things that I wanted you guys to pull back from, which was one of your first quiz questions, was organic molecule that is the source of energy currency, or is the energy currency, is adenosine triphosphate. And so what we're going to be talking about today is how cells are able to, and this is not necessarily just human physiology, this is actually just physiology. You'll see that pretty much any living being that needs to generate that form of energy currency, ATP, has to undergo cellular respiration. And cellular respiration is going to involve the breakdown of a metabolic substrate. And that substrate is glucose, which you're going to be seeing in just a little bit. That doesn't mean that glucose is the only energy we burn. We're actually burning quite a bit. Whatever you eat essentially is going to be broken down into ATP, whether it's going to be fats, whether it's going to be proteins. You'll be able to learn, especially in our digestive system, that we're typically protein sparing. We don't try and burn protein as a primary energy source. We typically rely on fats, and we rely on our carbohydrates to get broken down into that single monomer or single monosaccharide called glucose. And so the question that's going to be answered today is what happens to that glucose molecule. So let's take a look at our first set of slides. Cellular respiration. Eventually it's going to pick up. My computer's getting slower and slower nowadays, but eventually we'll pick up. And you'll see we're talking about cellular respiration. And what you see on my screen is not just a bunch of scribbling going on. That actually is the metabolic pathway. So if you were to draw chemically what exactly is happening with that glucose molecule, as well as the other things, our fats as well as our proteins, you can see that it's all completely metabolized. And if you've ever had instructors before ever draw it out, that's not the entire picture. In fact, they're giving you kind of an introduction. And again, I'm giving you an introduction here, but you would have learned this stuff in biology. I've heard of instructors talking about it, sitting there drawing this entire thing out. I'm not going to, and I want to give you a preface here, that I'm not going to require you guys to remember the chemical background. I'm not going to have you draw things out. I'm not going to have you guys draw where the hydrogen goes in glucose or anything like that. That's not physiology. That's not, at least from my opinion, that's not really the heart of what I want you guys to get across. So with that, again, this is mostly review. You guys are assumed to have known this stuff. This is really a refresher, but I feel is still an important background so that you understand how we generate energy. How do we generate ATP in order to fuel all those organ systems, to have them running? So with that, we're going to look at our topics today, cellular respiration, which we're going to break down into 3 components. We're going to see glycolysis, we're going to see the citric acid cycle, also known as the tricarboxylic acid cycle, also known as the Krebs cycle, and then lastly, we'll talk about the electron transport chain. But before we start, we do need to talk about the concept of metabolism. And what exactly is metabolism? You probably have heard the term, but metabolism is really just the whole concept of taking energy. It's the energy transformation in a cell. And we're going to be able to harvest energy from some source, at least harness a food source from some source of some kind. And we're going to be able to take that molecule, break it down, break apart those bonds, and transform that energy that we're getting from those bonds to couple that with other things. If you're wondering what things am I talking about, refer back to slide or your lecture number 1. And that last slide on there, I talked about all the cellular functions, reproduction, growth, movement, all that stuff is going to be involved. And this is really this sort of bouncing of energy back and forth. But when we break down metabolism, we can see that things are giving us energy and things are requiring energy. And so what we can divide metabolism down to, we can break it down to catabolic reactions and anabolic reactions. And these catabolic and anabolic, when we look at catabolic is the breakdown. We're taking large molecules. Whatever you ate today, for instance, if it was, let's say you ate potatoes, you took our starchy molecules, which is a long polymerized bunch of polysaccharides put together. We're going to be able to see glycogen later today. A string of glucose molecules, and we're going to break that down into smaller molecules. And we're going to take that glucose molecule, and we're going to break that down even more, and that's going to end up releasing energy. So catabolic means the breakdown of larger molecules into smaller molecules. The opposite of that is anabolic. Like I've used that term before, anabolic steroids. These are things that are building. So if we break down glucose into smaller molecules, we're going to be able to build larger molecules. And what molecules are we building? Well, we can talk about proteins again, just like we talked about in last Thursday's lecture. Or even last Tuesday's lecture, or the other day. The other day we talked about how we can produce proteins from amino acids. We start off with our primary sequence, we start to fold it in the endoplasmic reticulum, and then we're shipping those proteins out of the cell. All that requires energy that we're getting from our catabolic reactions. So here, anabolic means the synthesis of large energy molecules, large energy storing molecules, whether they'll be used for energy, whether they'll be used for growth, anything to support cellular functions. Now the other terms you see on here, exergonic and endergonic, again, something that I know we're just revisiting, but this is from the perspective of energy. And exergonic, if you remember, is the exit of energy, the release of energy. While anabolic is endergonic, meaning the energy entering. So exer and endergonic. Endergonic, this is energy going into in order to result in the synthesis of molecules. And typically when you look at these reactions, you have exergonic and endergonic reactions. We can now define these in terms of, when we look at reactions, you'll see that we're kind of ping-ponging back and forth between these two. We're going to see the term redox reactions. And let me help you kind of get into that. Before we get into what redox is, we have to understand the terms oxidation and reduction. Whenever we look at energy bouncing back and forth, and we're taking a molecule, we're going to be breaking it down, or we're going to be building it back up. It's going to be involving the movement of energy, and that energy comes in the form of electrons. So when we look at a molecule, whether it's going to be gaining an electron or losing an electron, are what's going to be the terms that are used, oxidation and reduction. These are actually the correct terms for them. So oxidation is the loss of electrons, while reduction is the gain of electrons. Now, don't think that oxidation is catabolic or any of that. This is just from the perspective of looking at what happens to the electrons themselves. Because the overall message, yes, at the very end is exergonic and endergonic, but during this process, when we look at a reaction, when we're looking at glucose, we're trying to see, are we stripping electrons off of it to use that energy to be used on another energy? Yes?
Is this the same process that removes hydrogen, or is that oxidation?
Exactly. So while I talk about the loss or the gain of electrons, typically when you're stripping an electron off, you've got an electron, but you're also going to be freeing, just like Eli said, we're freeing a hydrogen ion. So in essence, what this actually is, a hydrogen ion, it's a hydrogen with no electron, so it's a proton. So it's an electron and a proton. So typically these things go hand in hand. So the way oxidation and reduction is typically defined is a transfer of electrons. But I also like to just make it a little bit easier for you guys, is to also see the loss or gain of hydrogen ions. Whenever you see electron moves, hydrogen ions will go along with it as well. So the way you can remember a reaction, whether it's oxidation or reduction, is this mnemonic that I just drew out for you. Oil rig. Oxidation is the loss of electrons, while reduction is the gain in electrons. And I'll be able to give you an example in our next slide. And redox reactions are a combination. If you look at reduction, and ox for oxidation. And an example of that is this central dogma equation right here. What are we doing with our glucose molecule? This is glucose. This, you know, is oxygen. H2O is water. CO2. And then ATP. This right here is the central concept of redox reactions. It's a combination of oxidation and reduction. We'll be able to pick this apart. If you ever look at what's taking place here, glucose plus oxygen is going to get broken down eventually down the line. We're going to be able to get water and CO2. And then you'll notice one of our products on here. We know these are reactants. These are our products. And one of the products on there is ATP. And we're getting 36 ATP per glucose molecule. So my question for you guys is this an endergonic reaction or an exergonic reaction?
Exergonic.
Exergonic. How do you know it's exergonic?
Energy, ATP.
Because ATP is one of our products. Thank you. So we're getting a release of energy. And this energy can be used, that ATP can be used to couple with other reactions that are endergonic. Things that require energy. And everything that we're going to be learning as we go through neurophysiology, we're going to get into digestive, urinary, we're going to get into cardiovascular, the heartbeats, we're going to look at vessels expanding and contracting, all of that. Any type of muscle action, whether it's hormonal action, neural action of any kind, is going to require ATP. So this serves the foundation. So with this release of energy, we know that this is an exergonic reaction. And I'm going to erase this here just so we can read it. An exergonic reaction. And what we see is the net production of energy from the breakdown of glucose. Just like I said earlier, on the two main points that I talked about. And again, the redox reaction here, you can see my definitions on there. Now, this was the reaction. If you take a look at this reaction, and just kind of for fun, photosynthesis. Photosynthesis is the exact reverse of this. You take H2O, you take water and carbon dioxide, add a little energy, and you know that energy comes from the sun. And photosynthesis is the exact reverse, where your plants are producing glucose and producing oxygen as a byproduct. And you see what we're doing is we're doing the exact opposite. And then as another fact too, people like to think that plants are always producing energy, but they're only producing energy during the day when the sun is up and providing energy. But to support all the cellular functions such as growth, cells are also doing this. They're doing exactly this. They're taking glucose, the glucose that they were able to store, for instance, starch, like potatoes, storing those glucose molecules as a form of carbohydrates, as known as starch, to be able to store it when needed. So with this equation, we've known this equation for quite some time. The question is, where do these products come from? Okay? And typically when I ask this question of that CO2, where do you think CO2 ends up from? Or where does it come from? How do we get CO2 produced? Where do you think that CO2 comes from? By looking at our products over here.
From the glucose in the cells.
We'll get it from the cells, of course, but if you were to look at glucose, or you get it from oxygen?
Oxygen.
People like to think that it's from oxygen,
Glucose.
but that's not really true. And we're going to be able to answer that. We're going to be able to answer that question at the very end of today. So that same question that was asked was the same question they probably thought of when they first looked at this equation. And they knew that glucose plus oxygen produces water and CO2. You take a plant, you provide it or you take anything, you take cells and you provide it with those. It knows it produces energy. So the question is, where do we know, where does CO2 come from and where does H2O come from? So when scientists came up with that, they've said, okay, let's take a look at this. Let's take a cell or a plant or whatever it is. In fact, I don't remember what they used, whether it was an animal of some sort. But they can do this. Just remember when we talked in our first lecture, we talked about isotopes. They can take a radioactive isotope, and in this case, what they used radiated oxygen. So they take an oxygen that has radioactive properties, and now it's tagged. And what they did was they gave, let's start off with radioactive glucose. So here they used a radioactive isotope of glucose. Of oxygen in glucose. And they know if they give radioactive glucose with oxygen, where did it end up? Well, they ended up specifically with radioactive CO2. So they know that the oxygen on here came out in the oxygen in CO2. So they know during this whole transformation of turning glucose plus oxygen down that line in cellular respiration, the products, that oxygen came out tagged in the CO2. So they know this somewhere down the line becomes CO2. And then they did the exact opposite. To prove their point, they took radioactive oxygen, run it through that whole experiment again, find out where that ended up, and they found out they had radioactive water at the very end. And that's simple. You can just give somebody radioactive oxygen, and what they'll end up doing is either sweating it out or they'll end up urinating out water. Okay? So when we look at these reactions taking place, now that we're able to break it down, we can take a look at what we mentioned earlier, our redox reaction, and looking at our examples right here. Here we can see the example of oxygen, and oxygen is gaining hydrogen ions. So if we look at O2, eventually it's gaining hydrogen ions and also gaining electrons. So my question to you, is this reduction or is this oxidation?
Reduction.
It's reduction. And hence this is a redox reaction. We know that this, when we look at glucose, is losing hydrogen ions. So therefore it must be oxidation. hence why this is a redox reaction. So that they know. They know where the CO2 comes from. It comes specifically from glucose. And they knew that water comes in from oxygen. And what we're going to be able to do, and by the end of today, is be able to pinpoint where that takes place. So we know that oxygen creates water. And again, we'll point out where that takes place. So we're going to take a step back and go back to what we talked about in our biochemistry lecture, is to look at these reactions taking place. And view that from the perspective of, and you don't need to know the term Gibbs free energy, but this is the energy that you can measure from our reactants to our products. And this helps pinpoint the difference between exergonic reactions and endergonic reactions. Last week we saw this graph to our left. We see energy and we see time. And if we were to measure the energy in the reactants and measure the energy in the products, what we can do is we can measure the difference of energy in our products minus our reactants. And we know that the change in free energy, which is delta G, okay, and again, I won't ask you guys exactly what delta G is, but you should understand the concept that we're ending up with less energy than with what we started. And that must mean that delta G, which is the change is a negative change, hence why delta G is less than 0, that means energy is being released by exergonic reactions, which is exactly what the definition that I just gave you guys. And we know that the very bit right here is activation energy. It's energy that's required to get this thing started. So as we talked about, we knew in exergonic reactions that things can just happen. Eventually, maybe the reactants would eventually pick up enough energy that it would get over that little hurdle, which we know is activation energy, which you know from the quiz, and then the reaction would take place further. And in cellular respiration, we'll be able to see that. But in order to speed that reaction up, what do we employ? If you remember from last week's lecture, what do we do? What do we introduce into the system to speed this reaction up without changing the free energy? We introduce something that would speed the reaction. It's like it's something that catalyzes the reaction.
Enzyme.
Catalyst?
Enzymes. Catalyst, yeah, but enzymes are the reactions that is what I'm looking for specifically. Enzymes are introduced to speed this reaction up. And we're going to be seeing that in cellular respiration. We're going to be seeing a lot of enzymes that are involved to speed this reaction up. Okay, so we know energy is released. And if we look at endergonic reactions, if you were to measure the energy from reactants to products, you'll see that we're gaining free energy. Hence why delta G is greater than 0. It's positive. It requires energy. And again, same thing. You'll see energy is being introduced. And then we need a certain amount of activation energy to get to that hump to finally get our product. And where does that energy come from? It comes from our exergonic reactions. So exergonic reactions, again, are supplying the energy for endergonic reactions. So we break things down in order to build things back up. And then, as I mentioned earlier, our exergonic are our catabolic, and these are our anabolic reactions. Again, both of these comprising all of metabolism. So we couple these in order for cellular functions to take place. So now let's take that back to what we learned earlier in this reaction, and let's start to tie that stuff in. And keep in mind that since cellular respiration is an exergonic reaction, I want you guys to be familiar with this graph right here, because we're going to be able to look at that in just a little bit. But before we get into it, I want to go into the key players. Who is going to be involved in cellular respiration? We know glucose is going to be involved. That's a big giveaway. And then we need electron acceptors. If we're stripping electrons off something, electrons have a good amount of energy to them. And what we need to do with those electrons is we need some middleman to come in and carry that electron over down to its final place. And so keep that in mind. I know I'm kind of giving some vagueness right now, but that final destination, we need some carriers. And we have two carriers that are involved in this process. Nicotinamide adenine dinucleotide. And I would never ask you guys to remember the names of those. But we know they're called NAD, and the other one is called flavin adenine dinucleotide, also known as FAD. And both of these are actually derived from vitamins. When you learn about why vitamins are necessary, vitamins, derivative of B vitamin, it's niacin, also known as vitamin B3. And the other one is a derivative of a B2 vitamin, also known as riboflavin, so B2. So these, if you were to take a look, are electron acceptors, meaning that they're going to be gaining electrons, and then also they're also gaining the hydrogen ions that come with it. So you can see that we have NAD plus. It's going to be gaining a proton and gaining an electron, and therefore you're going to be able to see that NAD is going to be, if it's gaining an electron, is it being oxidized or reduced? Reduced. So we're going to see the reduction. Both of these are going to be reduced. Reduced to NADH and FADH2. So keep that in mind. These are going to be the key players, or middlemen, that are going to be carrying it over to their final destination. And then, lastly, adenosine triphosphate. This you guys already know because this was our quiz question. Adenosine triphosphate. And let's take a look at what adenosine triphosphate looks like. So top left, here you can see the structure of it. Remember, this was one of our nucleic acids that was specialized. We had adenine, which is our nucleic acid, our nucleotide. Ribose, which is our sugar, we had a pentose sugar attached to phosphate groups. And we have 3 phosphate groups, 1, 2, 3, hence the term adenosine triphosphate. And like I mentioned, there's bonds between each of these, and there's a lot of energy stored within the phosphate ions, or within the phosphate groups of that ATP molecule. And typically what happens with ATP is when you see ATP, we have our 3-phosphate, 2, 3. What we typically do to release energy from ATP is to break off that 3rd phosphate ion, or that phosphate group. And with that break, what we end up seeing is we see energy being released. We break down ATP into ADP, adenosine diphosphate. So that break off of that last phosphate ion is going to be used, the energy that's released is going to be used for endergonic reactions. This is an exergonic reaction. All this right here to the right is exergonic. Now the question is, what happens to that phosphate ion? Well, we're going to be able to see what happens with that phosphate ion. Typically what's going to happen is it's going to be passed along to the next molecule. And in that process, we'll talk about is on the next slide, it's called phosphorylation. Now, on the other hand, when we get ADP, how do we turn ADP back to ATP? And that's what's going to happen at the end of today's lecture. We're going to turn ADP back to ATP. After ATP is being used, it's going to be turned back to ATP. And that's where we're undergoing cellular respiration. We're harnessing the energy from glucose, combine that with a phosphate ion to turn that back into ATP so it can support cellular functions. And what cellular functions are they? Well, you can see 9 that I have on there for you, of which the ones that are relevant physiologically, start from the bottom, heat, flagellar movements, yeah, for sperm, sure, but not necessarily for bacteria. Cytoplasmic transport, oh, right there in the middle. Active transport, our sodium-potassium pump, if you remember that from last week, that was the other day, we talked about the sodium-potassium pump. We're pumping ions against its gradient, and that requires energy, and that energy comes in the form of ATP again. Importing metabolites, anytime we're ever moving things in and out, some things require energy, not all. Biosynthesis, of course, anytime any sort of anabolic reactions come to play, anytime we're building something is going to require ATP. Muscle contraction. Again, we don't really go over our musculoskeletal system, but anytime we get a muscle contraction to take place, it's going to utilize ATP to drive this myosin head and move that along. And then chemical activation. in order to activate something, activate an enzyme, activate a molecule, we're going to phosphorylate it, meaning we're going to not only harvest the energy from ATP, we're going to also attach that phosphate ion onto it. And that's how we're going to be able to tag that. You're going to see that numerous times today. So as we go through that process, when we're generating ATP, remember that we're tapping into ADP sources. ATP that's already been expended into ADP, we're going to combine that with the phosphate ion to replenish itself. So we constantly have this source going back and forth. So what does that look like? And I think you guys may not have this slide. You guys have it? No, but you will in a future slide. There's a small, all I did was take this small image and I kind of blew it up. And I believe it's well up there somewhere. But nonetheless, the real main message that I want you guys to get across from that is right over here at glycolysis. All I've done is take this image right here and blow it up. That's slide 13.
Yeah.
Okay. So the phosphorylation of glucose, all I wanted to do on here was to show you what's happening. With ATP, we're breaking apart that third phosphate group, harnessing that energy, and we're removing that hydrogen ion. So we're going to strip that hydrogen ion off that hydroxyl group, off glucose. So here what we're doing is we're removing an electron and removing a hydrogen ion. So if it's losing, this is the oxidation of glucose. We're stripping that hydrogen ion, and that phosphate ion that comes in from ATP is going to get placed onto where that hydrogen ion originally came from. So this is what we call glucose 6-phosphate. We've essentially taken a phosphate ion, we've tagged that glucose molecule, and that is going to be our first step in cellular respiration, which you'll be able to see in the next couple slides. So that is an example of phosphorylation. And what's required of this is a specific enzyme. Our enzyme on there is hexokinase, also known as glucokinase. That's the name of the enzyme that strips that hydrogen ion and attaches and metabolizes ATP and attaches that phosphate ion on there. And I'll talk about the relevance of that in just a little bit. So all of cellular respiration, when you saw that equation earlier, it's not like we take a glucose molecule and undergo one reaction and end up with our byproducts, our products. We have to go through several steps in place. And those steps that are involved take place in different areas of the cell. The first portion we're going to be able to see, if you look at cellular respiration, we're going to see is anaerobic, anaerobic. So the easiest way to define what aerobic means oxygen dependent. It requires oxygen in order for that process to take place. The other portion is called anaerobic, meaning it's oxygen independent, meaning that we can take our metabolic substrate, glucose, and we can burn that. And when I mean burn, I use the term loosely. We can have that undergo glycolysis, which is, if you look at the term, lysing is to break apart. Glyco refers to glucose. So we're burning, or we're breaking apart glucose. And where does that take place? It takes place in the cytoplasm. So if I were to draw out a cell, this is our cell membrane. And then we have glycolysis taking place. You don't necessarily have to draw that because I got that for you on the next slide over under the roadmap. Here you can see glycolysis taking place in the cytoplasm. And then in the presence of oxygen, we'll be able to undergo the next step. The term that I'd like you guys to be familiar with in glycolysis is that it is called substrate phosphorylation. Again, I will define what that term means in just a little bit. Aerobic, we're going to take that byproduct that comes in from glycolysis, which you'll be able to see in a little bit is pyruvic acid. We're going to turn that pyruvic acid into, we're going to shuttle that into the mitochondria. And this is a reaction called the transition reaction. We're going to take the byproduct from transition reaction, and which we'll know as acetyl-CoA, and it's going to undergo a cycle inside the matrix of the mitochondria. So we've shuttled pyruvic acid, we're going to shuttle that into the mitochondria. It's going to undergo the Krebs cycle. And then the last portion is called the electron transport chain, where we have enzymes littered all along the cristae of the inner membrane of the mitochondria. So these, if you remember in our on our lecture the other day, you saw a bunch of enzymes, a bunch of studded inner mitochondrial membrane, and that's where our enzymes are going to reside, and that's going to complete that last bit of cellular respiration for us. So like I said, you can see all this taking place in this general roadmap. And what we're first doing is we're taking glucose, we're shuttling that into the cell. If you remember, when we saw the other day, we knew that glucose, the concentration of glucose, is high on the outside. So we're shuttling glucose into the cell. And what we're utilizing here is this a carrier or a channel that we're going to be using to get glucose into the cell?
Carrier.
A carrier. And that carrier in this case is GLUT4. Its one task is to take glucose and move it down its diffusion gradient. Glucose is too big to cross through the cell membrane itself, so it's going to carry glucose from the extracellular side and shuttle it into the intracellular side. So we get glucose into the cell, and now that we get glucose into the cell, it's going to undergo glycolysis. And in glycolysis, we're going to be able to get ATP from. We're not getting all 36 ATP, we're just going to be getting just a little bit of ATP. So this is the first step in that process. And we'll be able to explain this a little bit, where you're going to see the reduction of NAD. And then our end product of glycolysis is pyruvate. And don't worry about that. If you're writing down what the end products are, it's cool. We're getting to that point. And then pyruvate, whether it's going to be present in oxygen or without oxygen, can go of two ways. There's a fermentation process, and actually the term isn't really fermentation. And I will get into that in just a bit. The other process is the aerobic metabolism, so oxygen present. So it is oxygen dependent. We're going to take pyruvate, shuttle it in. It's going to go through the Krebs cycle, also known as the TCA cycle, the tricarboxylic acid cycle. And we're going to release some ATP from there. And then we're going to use our key players, NADH and FADH2, that will contribute to the electron transport chain that happens along the inner membrane. And then that's where we're going to be able to get our bulk of ATP. So now that you get sort of the big picture, what we're going to do next is to zoom in to glycolysis. So let's take a look at our next slide in glycolysis. And this is another roadmap here, but you see what's taking place in the mitochondria. I've graded this out for a reason, so that we can specifically focus what's happening in the cytoplasm. And what you'll notice here is glucose is our reactant. Our product is pyruvate down the line. But there's numerous enzymes that are involved in every one of these steps. These are all byproducts. These are all midway points. And each midway point requires an enzyme. And I'm not going to require you guys to memorize those enzymes in between. But these are all the byproducts. These are all the steps that are involved to turn glucose back down to pyruvate. It has to go through each one of these steps. In fact, there's even more steps in these. This is actually just a simplified version. So what we know is the first bit. Glucose is going to get tagged with a phosphate ion by burning one of those ATP molecules. And we're going to turn that into glucose 6-phosphate, just like we saw on this slide over. So glucokinase is that first enzyme involved. Then the next step is going to be called fructose 6-phosphate. And then that next step, we're going to again use another ATP molecule and turn that into 2 molecules of glyceraldehyde 3-phosphate. Again, you don't need to know the names of these. But we've split that byproduct into 2 different parts. So if you take a look at that, what we're seeing is that we're using ATP. We're using specifically 2 ATP. Now the question that I'd like you guys to think about is, why are we using 2 ATP if we're trying to harness more ATP? Okay, so think about that. When we get into the next stage, once we split that fructose 6-phosphate into 2 glyceraldehydes, we're going to be able to see that NAD is getting reduced into NADH. We're stripping electrons off. And we're going to have our carriers that are going to pick up those electrons. Now the next thing you're going to see, we're going to have 1,3-biphosphoglycerate. Again, you don't need to know the names. But when we undergo the next step, you'll see that we're going to be able to phosphorylate our ADP. meaning we're turning ADP into 2 ATP. And then that last step, here we get phosphoenolpyruvate, and we're taking 2 ADP and turning that to 2 ATP. We're harnessing the energy as we transform that glucose molecule and turning that into 2 net ATP. So here we used 2 ATP, here we get 4 ATP. Our net gain from burning that one glucose molecule is 2 net ATP. At any point on here, did you see oxygen being used? Take a look through. Do you see oxygen at any point in here? No, we don't. Hence why it is oxygen independent.
So it's anaerobic, all of this,
It's
right?
anaerobic. Glycolysis is anaerobic. Now let's take a look at it from the what I mentioned earlier, is that we're using energy. This was an energy investment stage, and this was the energy harvesting stage. So let's pair that up with that free energy. Again, that graph that we talked about. And here you can see glucose. We use 2 ATPs to turn it into to glyceraldehyde 3-phosphate. So that product is right there at the very top. Glyceraldehyde diphosphates. And here you can see that by breaking apart those 2 ATPs, we're getting a gain in energy. So now I'm going to ask you guys to answer that question. That 2 ATP that we used, what would be the term for that? That ATP that's being used in order to end up with our product. If this is our reactant, this is our product, we're investing 2 ATP. It's just a little bit of energy that we need to invest in order for the reaction to take place. What was that term called?
Endoreactant?
What was that again? One more time?
Endoreactant? Endogonic?
Activation energy?
Oh.
That was a quiz question, right? Yeah, I remember the answer to that. So we're investing a little bit of energy in order for the reaction to take place. So that investment is the activation energy. Now, if you measure the amount of energy in glucose and pyruvic acid, that doesn't change. But we need to get this started. This was that example that I used earlier. In order for a reaction to take place, you need to invest a little energy. In order to get that roller coaster to go, you need to give it a little push. And that's exactly what we're doing here, starting that reaction. And of course, we have numerous enzymes that come in and take place. If you were to get glucose to turn into pyruvic acid on its own, you probably got to throw a lot of energy into it, and eventually it could turn into pyruvic acid. But we have actually 3 and even more enzymes that are involved that eventually will turn it into pyruvic acid with less energy. So enzymes, numerous enzymes are involved to make this as efficient as possible, to use the less amount of energy. If you can invest a little bit of energy and get this whole reaction to take place, great. That's exactly what we're doing. And that's what we use to invest. That's our payback. Because afterwards, we're going to be able to harvest it in these 2 points. As we turn these later on, you're going to be able to say that we get 2 ATP at this step and another 2 ATP at this step. And if you can see where that takes place, that takes place at these points, biphosphoglyscerate to phosphoglycerate and phosphoenolpyruvate to pyruvate. So that's what we talked about. And then another thing to note on here, the change in energy. We get a change in energy where our final products, pyruvic acid, has less energy than what we end up with glucose. So here we would note that the change in free energy is going to be negative. It's less than 0. So think about this conceptually. If I have less energy in pyruvic acid, where did that energy go? I lost a good amount of energy. That's delta G. Where did that energy go? I'll give you a clue. What you learned last week, you learned that that came off as heat. Kinetic energy. That kinetic energy is off to do something else. We're not giving off heat necessarily per se in this process. Where did that energy go?
Go to next step.
Let's do potential energy.
What did we end up with? What was our product?
Pyruvate.
Okay. what was our net gain in energy?
2.
2 ATP. So in order for that ADP to get turned back into ATP, its high energy state, we're turning glucose, putting 2 ATP into this process to then release the energy from those byproducts to turn ADP back to ATP. And that ATP can go back and support all of these processes. And once they get used, when we build that protein, we're going to end up with a bunch of ATP that's now back to ADP. We need to turn that ADP back to ATP. And in the presence of, without oxygen, we take that glucose molecule, do exactly that, and we end up with 2 net ATP. Does that make sense?
Yeah.
Okay. So that's where that energy went, is the production, the phosphorylation of ADP back to ATP so that it can do work. So this process right here, glucose to pyruvic acid, is that an endergonic or exergonic reaction? How do we define ender and exergonic? The direction of energy, right?
Yeah.
And this is energy exiting the system.
Exergonic.
Okay. Now ADP to ATP, is that exergonic or endergonic?
Endergonic.
It's endergonic. It requires energy to turn ADP back to ATP. So this is another example of where we used an exergonic reaction, coupled that with an endergonic reaction to take place so that we have our source, our energy currency right here, ATP. That's exactly what we're trying to achieve. So now, out of the 36 ATP that we learned from the very beginning, this is where we get 2 ATP first. So it requires a little bit of energy at first in order to end up with that. So let's take a look from the perspective. We've talked about the energy perspective. Now let's just look at what's happening to the hydrogen ions and the electrons themselves. And the breakdown of what we started off with glucose. Glucose is C6H12O6. There's 6 carbons. And eventually when we look at pyruvate, it gets broken down into 2 pyruvate molecules where we get a pyruvate molecule with C3H4O3, and it's 3 carbons. So we have 2 acids that we end up with, each with 3 carbons. So if we look at the carbons, then eventually we have to look at the hydrogens, and we have to look at the oxygens. And this, if you remember from chemistry, is what we call stoichiometry. All we're doing is making sure, do we end up with the same carbons at the other end? 1 C6 for glucose, and we end up C3 plus C3. So we have 6 carbons on both ends. So our carbons are good, right? What about the hydrogen molecules? No. Okay. What about the oxygen molecules?
Yeah.
Yes. So they all balance out. So we're losing 4 hydrogens in this process. So the question is, where do those 4 hydrogens go? And then, consequently, what happened to those electrons that follow those hydrogen ions? That's the question.
NAD.
And that's where our middleman comes in, because we can't just have electrons just floating around. They've got to be carried by somebody. And that's where we first see our NAD. Our NAD come in. Okay, so each NAD that comes in gets picked up by, each electron that comes in, we get a pair of hydrogen ions and a pair of electrons. And that gets picked up by each NAD molecule. So each NAD carries 2 electrons and 2 hydrogens. So we get, with this process, when we go from NAD plus an H plus 2 electrons, we end up with NADH plus AH plus. And that's okay, because the main idea of what we're getting across is that we get, in this process, we're reducing NAD to become NADH. It's our carrier. We'll talk about where that NADH goes. So now that we can continue, let's continue. We've already talked about our glucose transporter, as we mentioned in this previous slide, our GLUT transporter. What we're doing with that transporter, once that glucose is transported in, what we're doing is we're going to be activating that glucose. Take a look at that first step on here. Glucose to glucose 6-phosphate. This is where glucokinase or hexokinase, as I mentioned in this previous slide, hexokinase is coming in to phosphorylate glucose. Now the question is, why do we need to do that? So we're activating it, and in the process we're activating it with a phosphate ion that comes in from ATP. And through that phosphorylation, what we're doing is we're trapping that glucose into the cell. Remember what I said about that phosphate and oxygen. They have a tendency to become very electron hungry. And if it's electron hungry, if you take a look at it, this is going to make it more polar. And therefore, if it's more polar, not only do you have a large molecule, but now it's even more polar. So it traps it into the cell. So that phosphorylation of glucose starts the process of glycolysis. So now that it's not able to leave the cell, I do want to throw in this next slide on here. This is just going to kind of help clarify things. Because when we talk about pyruvic acid and pyruvate, you'll probably hear pyruvate, you'll probably hear pyruvic acid. The book uses one, you read other sources and they say the other. And you're probably thinking, what's the difference between the two? Well, one, pyruvate is just, all it is the ion. You've taken the pyruvic acid, you see 1, 2, 3 carbons. Here's our carboxylic acid right here. This was our functional group. And all we're doing when this is called pyruvic acid because it has an acid, what we're doing is taking off that excess hydrogen and we're calling it pyruvate. So that's the name of the ion. So this anion is called pyruvate. Remember, anion is negative, cation is positive.
So it's just adding, what was it, a hydrogen?
Yeah, so if it was an acid, it would have that carboxylic hydrogen on there. This is called pyruvate just because we stripped it all off. That's the only difference between the two. But basically, they're the same thing. One is ionized, this one's electrically neutral. But pyruvate and pyruvic acid are the same thing. This right here just basically does the same thing that we've already looked at. Except again, from a different perspective. We're taking glucose and we're splitting it into 2 pyruvic acids. Again, all the different intermediates or byproducts in between. We're using 2 ATP to turn it into fructose 1,6-biphosphate, and then we take each of these, turn them back into their intermediates, eventually back to pyruvic acid. So here you can see at each step where ATP is being harvested and where ATP is being invested. So this represents the activation energy. These 2 points right here represent the the energy harvesting stage. And these 2 are our progress. So I've used that term earlier. If you go back to your earlier slides, I called glycolysis, in the process of glycolysis, it's called substrate phosphorylation. And in the book and in many other sources, they have you explain the difference between substrate phosphorylation versus oxidative phosphorylation. So you should be able to answer that by understanding what exactly I mean by substrate phosphorylation. And what we're doing is we're taking ATP, we're stripping off, we're harvesting the energy from ATP back to ADP. We're taking that phosphate ion and we're stripping that back and putting it back to ATP. And let's take a look at how that works. Let's take one of our substrates. And here I'm taking a look at phosphoenol pyruvate. If I take phosphoenol pyruvate, you'll see that I'm going to turn that into pyruvate. This is our reactant. This is our product. We're going to have an enzyme. And that enzyme on here is called pyruvate kinase. We're going to be able to strip, we're able to take phosphoenol pyruvate and turn that to pyruvate, and then also harness that energy from that process to turn 2 ADP to 2 ATP. So let's take a look at what that looks like from a molecular level. Here's our enzyme. This right here is a site that is active. And what would we call that site? Active site. Right? So this is the active site. It's an active site because it has our substrate, and our substrate in this case is phosphoenol pyruvate. You take that substrate, and here you can see the design of it. You can see you can see the makeup of it. 1, 2, 3 carbons. Here's our 3 carbon substrate. You see a phosphate ion that's attached to it. So in this active site, we bind PEP. The other substrate that's required, so if this is reactant number 1, we need reactant number 2. They should start to look familiar, because if you remember, we did A plus B turns to C. You guys remember that?
Yeah.
Right? And in that process, what we're doing is we're also releasing energy. So reactant number 1, reactant number 2. And what's happening in this process, this enzyme has one job, and that is to, when these two reactants bind, is to strip that phosphate ion and catalyze the reaction to bring that over to adenosine diphosphate. So there's that phosphate ion, it gets transferred over back to ADP, ADP turns to ATP, and we end up with our final products, pyruvate and ATP.
So basically, the phosphorus went to hang out with the whatever.
Adenosine, yeah, diphosphate.
And that's how ATP was turned?
Yep.
It's just the switching of the phosphorus?
That's all it is.
I thought it was the loss of it.
So what we're doing is we're metabolizing that substrate, hence the substrate, PEP. You can even get this with the same thing if you were to look at fructose 6-phosphate. There's an enzyme that will, biphosphoglycerate. You can take 1,3-biphosphoglycerate, and that's going to undergo, this is the substrate, it's going to undergo a process where another enzyme, very similar to this one, catalyzes the reaction of taking that phosphate ion, pulling it off of this, and throwing it back to adenosine diphosphate. And again, where does that energy come from? That energy comes from that energy transfer that goes back from, again, going back from this step to this step to this step, that energy right here that's lost is coupled to drive this enzyme. So that enzyme right there, that phosphorylation of ADP turning to ATP requires energy. So the breakdown from PEP down to pyruvate fuels that. What questions do we have? Good. So now that we know the active site, and again, I'm going to go into a little bit more detail that you don't necessarily need to know. We know that this is the active site. And you knew from last week's lecture that if I start supplying more PEP and more ADP, this is going to drive this reaction forward this way. So one of the things that we could modulate this enzyme to work a little bit faster, instead of working on the active site, we were talking about modulating it from the active site. Let's talk about working on another site that's not the active site. Do you guys remember the name of the site that's not the active site?
Allo.
You guys had a quiz recently, right? I hope you guys were studying. There's a
Allocation.
name on a site of an enzyme that's not the active site. Allosteric site. And we had an allosteric inhibitor, and we had an allosteric competitor. Oh, no, I'm sorry, activator. Allosteric activator and an allosteric inhibitor. So an allosteric activator is something that would activate this enzyme by binding to the allosteric site. Well, naturally, if you want to turn on this enzyme, wouldn't it make sense that you would have one of these byproducts? One of these byproducts early on. Imagine this, if all of a sudden you get, let's say you get fructose biphosphate. Where is it? Fructose, you actually don't see it on here. But one of the metabolites on here is fructose biphosphate. If that comes in and turns on this enzyme that's required over here, wouldn't it make sense? If I'm producing a bunch of one of these intermediates, hey, ramp up those other enzymes down the line because I'm going to get them to work harder. So that one of these metabolites comes in, turns this enzyme on, and now this enzyme can work. You're starting to activate more of these enzymes so they can work later on the line. Now, think about what's going to inactivate this enzyme. Turn this enzyme and make it work slower. Well, one of our products in this is ATP. ATP can come in and phosphorylate this enzyme to turn it off. And that should make sense, because if I'm starting to get a bunch of ATP, I don't really need glycolysis anymore. I've got enough ATP to do what I need. you're not just trying to produce a bunch of ATP and just say, hey, produce a bunch and store it. You only use what you need. So here's that concept, that if I need more ATP, I'm going to make it. And if I have enough ATP, I'm going to turn it off. That sounds like some sort of feedback mechanism, doesn't it?
Yeah.
Which one is that? Is that positive or negative? It's negative feedback. So ATP comes in and turns off this enzyme so that it can't make more ATP. You're going to see a lot of that in physiology, especially when you get into your careers later on. You're going to learn that these come back to turn it back off. This is all an example of negative feedback. And not one that I require you guys, that was sort of a bonus. I'm not going to require you guys, I'm never going to say that on the test, what's going to be the allosteric inhibitor of our pyruvic kinase. That's cool. That's way too much information. You get on later on, medical school, sure, yeah, you're going to have to know that. And then also, what you would also learn, especially even in pharmacology, you'll learn that these enzymes that are involved, something's got to turn off that enzyme to stop that production of something. If you make too much of something, which is typically what a disease, or you don't make enough of something, a disease is exactly like that. So maybe we just got to turn that enzyme off a little bit. We'll cover that when we get into our cardiovascular lecture. There might be something that's happening, so what we need to do is turn something off. We need to turn an enzyme off. Therefore, we'll work on that allosteric site. That's exactly how these drugs work. If you learn this stuff, you really learn how drugs work, and you learn on a molecular standpoint how that works, you're going to make some big money, because that's exactly what pharmaceutical industries need. They need people to understand pharmacokinetics. They understand how the molecules come in to turn an enzyme off, and what's that going to do to the other systems? Is it going to affect anything else? Are they going to produce the unwanted side effects? Exactly. Okay, so I'm digressing. I don't want to get too into it, but at least help you stimulate some things. All right, so that's substrate phosphorylation. Now, what we end up with at the end of glycolysis is pyruvate. And the question is, what do we do with that pyruvate or pyruvic acid? We want to convert that over into our next product called acetyl-CoA. So we've taken pyruvic acid, and you can see that in the reaction on the next slide over. We take our pyruvate, and we want to shuttle that into the mitochondrial matrix. We need to get that into the matrix. We started off, that pyruvate was produced in the cytoplasm. It's going to get shuttled into the mitochondria, and in that process we need to convert it into acetyl-CoA so that it can make it into its next step, called the Krebs cycle. So here you can see this process. We're going to take pyruvic acid, or pyruvate, and then it gets shuttled into, here's our outside of the mitochondria, here's the inside of the mitochondria. And this is going to land itself, taking a look at that pyruvate, you're going to see that pyruvate, we're going to strip off this carboxylic group, and if you look at it, C and then the two oxygens, conveniently that's going to become CO2, right? So there's our first product of CO2. Figure that, didn't that come up from a sugar? You guys thought it was from the oxygen. No, we get our first CO2 come off. We strip that carboxylic group. We're going to reduce NAD into NADH. And then we're going to have the byproduct from Krebs cycle. So this coenzyme A comes from the Krebs cycle. Oh, I'm sorry, no, sorry.
So the H
Just kidding,
was just hanging in the mitochondria?
What's that?
The H was just hanging inside the mitochondria?
That H?
Yeah.
You'll learn in just a little bit. We'll revisit that question in just a little bit. So now what we're going to be doing is we're going to get a coenzyme A. Coenzyme A is going to come in, that's coming in from the mitochondria. That will come in and become acetyl-CoA. So these two carbons that we saw from pyruvate, we've stripped off that carboxylic group, which becomes CO2. We're going to attach acetyl-CoA. Attach coenzyme A to become acetyl-CoA. Because acetyl-CoA, in this process, you'll see the hydrogen. We'll talk about the hydrogens in a bit. And of course, the question becomes, what happens to that CO2? How do we get rid of that CO2? Well, that CO2 gets shuttled out of the mitochondria into the cytoplasm. That cytoplasm makes its way back out. We're going to shuttle that back out of the cell. That cell is going to get made into the outer extracellular fluid, of which part of the extracellular fluid is the capillaries. That's coming around delivering fresh fluid to the cell. So that CO2 gets diffused out into the circulatory system. And where does that make its way to? To the lungs. This slide, I keep wanting to remove this slide. It's basically the same thing. I'm just kind of beating a dead horse here. It's glycogen to glucose, glucose to pyruvate, pyruvate back to acetyl-CoA. So what I've drawn on that last slide is from pyruvate to acetyl-CoA and CO2. Oh, no, I know why I didn't want to remove this slide. The other two things on there, amino acids and fatty acids, I mean fats and proteins, you'll see that enter the process here too, through acetyl-CoA. They can get converted to acetyl-CoA, so they can get fed into the citric acid cycle or Krebs cycle. Mm-hmm. That is.
Where does that come from?
So this is coenzyme A. This portion right here,
Yeah.
coenzyme A. That comes in from the mitochondria. And you have to see the reaction taking place to see where that comes from. So now that we have acetyl-CoA, we're going to take acetyl-CoA and it's going to merge with oxaloacetic acid to form citric acid. That portion right there, you can see on our next slide. So here's acetyl-CoA. This is what came in from the transition cycle, acetyl-CoA. This cycle spins around. This entire citric acid cycle spins around, and one of the final products is called oxaloacetate, or oxaloacetic acid. That comes in to bind to acetyl-CoA along with a water molecule to form its byproduct, citrate. So keep in mind that this cycle constantly spins around. And it goes over and over again. As long as you're shuttling in acetyl-CoA, it's going to continue. So when you get oxaloacetic acid to bind to acetyl-CoA, you're going to see it's actually going to pull off that coenzyme A. So that question of where that coenzyme A came from back here. This is just an intermediate. It's one that just comes right back in to feed it right back. It forms its intermediate called citrate. So here you can see 1, 2, right? You see 1, 2 carbons. So that becomes a 4-carbon called citrate. And we call this the tricarboxylic acid cycle or Krebs cycle. But tricarboxylic acid cycle because we have three carboxyl groups. 1, 2, 3. And that's what we start off with. And each of these steps, and again, you're going to see a bunch of intermediates. The question is, do you need to remember them? No, it's okay. You don't need to know them. But the ones that you're going to see that you could remember, if you remember from biology, the mnemonic that I remember using is GPAXMO. And I remember this because it was a stupid mnemonic, but you know what? I can remember it. Glucose, pyruvic acid, acetyl-CoA, citric acid, alpha-ketoglutarate, succinic, malic, and oxaloacetate. And I still remember to this day because of that mnemonic. And each of these are the intermediates of the citric acid cycle. And again, you don't need to know each of the steps, you don't need to know the molecular structures, but what you can see from here is citrate turns into isocitrate, and you'll see that this mnemonic doesn't cover all the intermediates in before that. And actually, there's a lot more intermediates than that. This is, again, like a high school biology class that I took. If you look at one of any of these carboxylic groups, you'll see that we strip them off. And that stripped off carboxylic group turns into CO2. So right here, you can see this is our first CO2 that's pulled out. Where do we see the next CO2 pulled off? What we're doing with this right here, these two C's, we're pulling off another carboxylic group and turning that into the next succinyl-CoA. So we have two CO2s that we've pulled off per turn of the cycle. Here's succinic, here's fumarate, malate, oxaloacetate. And then once that last metabolite comes in, it binds to another acetyl-CoA and repeats the cycle again. Another acetyl-CoA gets shuttled in, it's going to turn again. So as long as, like I said earlier, as long as you're feeding acetyl-CoA into the system, you're keeping the system turning over and over and over again. And in this process, per turn, what we're doing is we're just counting each of our byproducts. CO2, we definitely want to look at ATP, and we also want to look at NADH, and we also want to look at FADH2. CO2 and ATP should be pretty obvious. These were our two electron acceptors that were reduced into NADH and FADH2. So let's just look at per turn of the cycle. Let's count each of these. How many CO2s do we have? Take a look through.
2.
Just 2 CO2s, right? Do you see ATP anywhere on here?
Yes, right there.
Yeah, right at the bottom. So here what you're looking at is, it's actually GTP, guanine triphosphate. It's okay, it's another form of ATP. Don't want to get into too much detail. But what we're doing is we're generating one ATP. NADH, where do you see NADH on there? 1, 2, 3, cool. And then how many FADH2 are you seeing on here?
1.
Just 1. That's per turn of the citric acid cycle. So, my question to you is how many turns are there per glucose molecule?
2.
2. How'd you guys know that? Well, hopefully not because you just said it right there. I realized that after I asked that question. But, yeah, well, that glucose molecule is broken down into 2 pyruvates. Let me go back to an earlier slide. That glucose gets broken down into 2 pyruvate acids. And even going back to this earlier slide, that glucose molecule gets broken down into 2 pyruvic acids. So that pyruvic acid gets converted to acetyl-CoA. That means per glucose molecule, it's going to spin through twice. So let's go back to that earlier thing that we pointed out, where we summed it all up. And we're now going through 2 turns of this. Times 2, times 2, times 2. And that means we get 4 CO2, 2 ATP, 6 NADH, and 2 FADH2. Don't worry about writing that real quickly, because you know what? I summed it up for you on the next slide. Oh, and one thing also too on here is H2O. H2O is introduced to the system. So every time you have H2O, every time you undergo a Krebs cycle, you do use H2O. That's not really a big point, but I just wanted to mention that. So now summing this all up, we've now looked at glycolysis. We looked at the transition reaction. We looked at the Krebs cycle. We know that glycolysis ended up with 2 net ATP and 2 NADH. Transition reaction, we saw 2 NADH and 2 CO2. Again, per pyruvic acid to acetyl-CoA. And then Krebs cycle, we saw 2, 6, 2, and then 4 CO2. Just as we were able to sum that all up over here. So if we're going to go in, what's our net ATP right now?
4.
2 plus 2.
4.
4 ATP. We get 4 ATP in this process up to this point. But I thought we were making 36 ATP. We've only made 4 so far. So the question is, what happened to that? Where are those other 32 ATP coming from? That's where we have the next step. The next step being the electron transport chain. So our final destination for the electron transport chain for our products at the very end, we were able to see on here the conversion of those carbons from that acetyl-CoA getting transformed back and forth. And it's going to continue on this cycle over and over and over again. So the fate of that glucose molecule is done. It's not being used for anything else. We've stripped off the carboxylic groups off each of these intermediates, and we've pulled off that CO2. If you were to sum this all up, sum up all those carbons. We know glucose started off with 6 carbons. We get 2 carbons from 2 CO2 produced in the transition cycle. We get 4 carbons in the 4 CO2s that are produced in the Krebs cycle. Those carbons are done. What we ended up with a bunch of is NADH and FADH2. We've ended up with a bunch of electrons that are carried by NADH. And not just the electrons, also the hydrogen ions. So this is where they come into play, is the electron transport chain. Fancy that. I was just talking about a bunch of electrons. Now we're going to transport it. And so now we're looking at the cristae, the inner membrane of the mitochondria. So the inner membrane is littered with a bunch of enzymes. And if you were to take a look at it and actually dissect it and look at it, you can see it's just studded with a bunch of enzymes. Those enzymes, those mitochondrial enzymes, represent 5 major complexes. NADH hydrogenase, actually dehydrogenase. NADH dehydrogenase, succinate dehydrogenase, which is also known as CoQ10. If you've seen CoQ10, you've probably seen at health stores, you would have seen something called coenzyme Q, CoQ10. I know that I've seen it at Costco. They were trying to sell you on buying this stuff, and they're like, oh, it raises energy levels, and it does this and that. No, it doesn't. It's a byproduct in the electron transport chain. You already have enough of it. And that's just kind of another thing that you think about when you think about supplements they try and sell you. Your body produces it. They're just giving you more of it. If you have more of it and you don't need it, you eliminate it. So where that money go to? You end up pissing it out. So don't worry about that. So succinate dehydrogenase. And of course, if your doctor says you need to take a supplement, take the supplement. Don't ever say, Marlon told me I don't need to take it. Not a doctor. I don't let them determine your medical or your course of treatment. But nonetheless, in normal people, you don't need additional CoQ10. The other thing you're going to see on here is cytochrome BC1. There you'll see something else called cytochrome C, which is not necessarily a complex, but is also involved in this process. And the next complex 4 is called cytochrome C oxidase. You don't really need to know the terms of these. It's okay. Complex 1, 2, 3, 4, 5. I get it. The 5th complex, on the other hand, is ATP synthase. And right off the bat, if I were to ask you, what do you think ATP synthase does? We know it's an enzyme, because it ends in ACE, just like all these other enzymes end in ACE. What do you think ATP synthase does? Synthesizes ATP. So that's our final step. So each of these right here, 5 major complexes, are all in this process of synthesizing ATP. But technically speaking, it's that last bit. All these other, in the electron transport chain, are going to do exactly that. They're transporting electrons through these major complexes. So we can see that on this next slide. It's a little bit of a dated slide, but at least you get the idea of where we're at. Citric acid cycle is happening on the inner fluid area of the innermost membrane of the mitochondria. On the outer layer, this is the outer membrane, the outer layer of the mitochondria. And in between is the inner membrane space, in between the inner and outer mitochondrial membrane. And all along the inner mitochondrial membrane, you'll see 1, 2, 3, 4, and then 5th are ATP synthase. These are the enzymes that you saw on this slide right here. So to take a look at what's happening is we have to see the process that's taking place. And each of those steps are listed on here, so at least you can see what's taking place. So if you'd like to follow along, I can describe what's happening. The first complex is called NADH dehydrogenase. If you look at the term dehydrogenase, what do you think NADH dehydrogenase is doing? It dehydrogenases.
Removal of water.
Right.
Remove hydrogen.
Remove the water.
Yeah, it removes, well, not necessarily with water.
Hydrogen.
It's removing a hydrogen. And where is it moving the hydrogen from? NADH. So if you're removing a hydrogen, you're also removing an electron that comes with it. So we get an NADH, and NADH carries 2 electrons and also carries 2 hydrogens with it. So what we're doing is if we're taking NADH and we're losing electrons, are we reducing or are we oxidizing? We're losing electrons, oil ring. Oxidation, right? We're oxidizing. So we're losing electrons. We pull the electrons off that NADH. Those 2 electrons get handed to the first complex. And what we're doing when we take that high energy electron, we're going to pass it through. And it's going to get passed over to coenzyme Q, also known as CoQ10, also known as succinate dehydrogenase. And that's going to hand it over to complex 2. And complex 2, actually actually no, I'm sorry, complex 2 is succinate dehydrogenase. It's going to now get carried over to complex 3. And when we get complex 3 coming in, we should be able to see it on here. Complex 3 is going to take that electron, and it's going to pass it over to cytochrome C, which is going to now hand it over to complex 4. So what we're doing in the electron transport chain is we're taking these high energy electrons that came from that NADH, and we're passing it along like hot potato. It's got a lot of energy. We transfer it over to the next step. Then transfer it to the next step, and we're removing those high energy electrons, and we're removing a little bit of energy from it in that process. What are we doing with that energy? Well, we're driving hydrogen ions across. Every time you have an electron pass over, you're going to allow a hydrogen ion to pass through. And it's going to pass from the matrix into the intermembrane space. So we're pumping it across into an area. And if you notice, what we're doing all along these complexes is we're pulling hydrogens against its concentration gradient. I have a couple slides down the line called the chemiosmotic mechanism. This is exactly what I'm describing here. So in that process, I'm passing the electrons through, and in that final step, we get at complex 4, we're driving our third hydrogen ion across. What do we do with those electrons that get left over at complex 4? We have these electrons. We got that electron. That's at that last step. We got to do something with those electrons, and that's where the electrons combine with 2 free hydrogen ions come in and meet that oxygen molecule. Where does that oxygen molecule come from? How do you get oxygen? You kind of breathe it in, right?
Breathe. Yeah.
That's my answer. That oxygen comes into the system, picks up that electron. So that oxygen ion, we take that oxygen, we split that oxygen, and that oxygen is now an electron acceptor. So if that oxygen is gaining an electron, is it being oxidized or reduced? Reduced. So the reason we take in oxygen is so that it can be reduced into H2O. Right? In other words, we're taking that oxygen, it's picking up 2 electrons, and if it's picking up 2 electrons, it's got to pick up those 2 hydrogens. So 2 hydrogens and half an oxygen molecule become H2O. So if the question is, why do we breathe oxygen? The sole purpose is the second to last step on the electron transport chain. All it does is it picks up those high energy electrons. That's all it does. Because remember what I said from the very beginning. Oxygen and phosphorus are very electron hungry. So they pick up the electrons at the final end. Okay.
I just want to make sure I understand it. So NADH is a carrier that goes into every complex, right?
NADH, what it's doing is it's actually stripping, the first one is called NADH dehydrogenase.
But you had said that the hydrogen had 2 electrons, but they're losing an electron each, right?
Yeah.
But where do the rest come from?
I'm sorry, one more time?
You said it had 2 hydrogen electrons when it came in, right?
So NADH, it's typically NADH plus a hydrogen ion.
And then it starts losing those hydrogen
Yeah,
ions.
and it pulls those hydrogen ions, one of which that, two of which, those hydrogen ions get shuttled across.
Uh-huh.
Okay? And then we have hydrogen ion, we have the 2 electrons that come in with it. So the dehydrogenase pulls those electrons from NADH and passes it along complex 3 and complex 4. And each of those steps is pulling a hydrogen across into the intermembrane space. Did that help?
Yeah.
Now the question is, what do we do with that NAD+? What are we going to do with that? We're just going to be like, okay, cool, you're done NAD+, go ahead and go die off somewhere. No, we recycle it. That goes through the step, every other step in glycolysis, in transition cycle, as well as the Krebs cycle. Every one of those steps that needed NAD+, it gets recycled in that process. And so we continue. So now we've left off, we've got our water molecule, we're now gotten rid of our oxygen molecule. We have hydrogens now in the intermembrane space. The question is what happens also to FADH2? And FADH2 makes its way into complex 2. It actually bypasses complex 1, because complex 1 is called NADH dehydrogenase. It's not called FADH dehydrogenase. Complex 2 will do it. It only takes complex 2, so it's only driving FADH2. It's driving electrons through complex 3 and complex 4. Don't worry about that. It's written down a little bit down the line. So all of that summarizes everything that I've just talked about. This portion right here talks about complex 1, 3, and 4, and shows you the free energy per electron. And that's just showing you that these electrons are very high energy electrons, and they get passed through. 1 down 2. And that energy that's freed up from complex 1 to 3 and from 3 to 4 is being used to drive that hydrogen ion across into inner membrane space. So that, in the inner membrane space, is called the chemiosmotic mechanism. And this is what we're doing where we pump those hydrogen ions into that space. So we formed a gradient. And as I said, it uses energy that we saw from those complexes to throw it all up into the inner membrane space. In that process, we talked about NADH. We transfer three hydrogen ions through complex 1, 3, and 4. FADH only transfers through 3 and 4. So with that, we know that FADH only transfers 3, FADH transfers 2. So with that being said, now that we've had all of our hydrogen ions, we have to take a look at our fifth complex. And that fifth complex, again, is ATP synthase. And what that does is it uses that... I totally forgot about a break, guys. I realized that. I apologize. I got way too into it. Okay, ATP synthase is what that's doing is the ATP synthase. It's using that chemiosmotic gradient. It's using those hydrogen ions all the way up to the very top. I totally apologize. I totally did. Not only is it a break for you guys, it's also a break for me. And I realized I did not take a break for myself. Hydrogen ions. I was wondering. I'm like, why am I like so tired right now? So those hydrogen ions are going through, and they're going to make their way down this gradient. Okay. So hydrogen ions, every time it passes through, it's going to cause this whole ATP synthase to spin and create. What is it creating again? What is ATP synthase synthesizing?
ATP.
ATP. So it's going to turn ADP into ATP. So ADP plus a phosphate ion. So if you were to look at this ATP synthase molecule, or this enzyme, its two substrates, or two reactants are ADP and phosphate ion, and it's using this gradient. It literally is using all these hydrogen ions that will float through. It's using it like a waterfall. It's exactly what it's doing is, as hydrogen flows through, it's using that gradient to couple that to phosphorylate ADP to turn it to ATP. And every time a hydrogen ion flows through, it phosphorylates an ADP to ATP. So knowing that earlier, as I described, if NADH transfers 3 hydrogen ions and FADH transfers 2 hydrogen ions, we know that each NADH molecule produces 3 ATP. And each FADH2 produces 2 ATP. So this is actually a really cool molecule, because if you take a look and see how it's spinning, all of these right here are subunits. So this, you're seeing in its quaternary structure. Each of these right here are tertiary structures that are folded up. Take a look at the outside. These, on the outside, are hydrophilic groups. On the inside, hydrophobic groups. Each of the sides that are exposed to water are hydrophobic. And then you see the entire configuration of this. If you were ever to study the molecule itself, it spins exactly this way. So that is oxidative phosphorylation. And if you look at the term oxidative phosphorylation, all we're doing when we discuss the term oxidative, and I do have a slide that talks about that. Oxidative phosphorylation, please tell me. Please tell me. Okay, I'll describe that for you. Oxidative phosphorylation is there it is. Oxidative phosphorylation. This is that process right there. Is what we're doing is the oxidation of NADH to NAD+. This process, where substrate phosphorylation, we use a substrate. Here, we're oxidizing NADH to become, to help phosphorylate down the line in ATP synthase to phosphorylate ADP. So that is, let me just kind of quickly sum all that up for you right there. 3D image of protein folding, and I already talked about that. This last one right here is something that helps put this together for you. So we'll take a break after this. In the interest of time, can we take a 5-minute break after this? Is that okay? So this is going to help summarize exactly what I just talked about. And in fact, I just posted this on Google Drive too, so you can get a copy of this as well. It's also on Canvas.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. The electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates a gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, 2 mobile carriers are also involved: ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and beyond, which combine to form ATP. At the start of the electron transport chain, 2 electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of 1 hydrogen ion for each electron. Next, the 2 electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. 1 hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation. This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
Was that at least helpful? Yeah. Okay. I'll take a 5-minute break. Get you guys back at 9 o'clock and we'll sum up